Hello, everyone. Welcome to the second webinar series, The Road to ICOBA. And why we chose today, 26th of March, because we want to highlight the World Parasitic Awareness Day. The World Parasitic Awareness Day actually is quite young. It has been sorry, I can see some marks, but it has been established last year for the first time, and this is the second year for the World Parasitic Awareness Day. It has been selected to be 20th of March because it will coincides with the winter to spring, the start of the spring where parasites flourished, same like flowers and so on. I'm not sure how to remove this. Yeah. So what is ICOBA and what is World Federation of Parasitologists? I am Hissa Al-Farsi and I am a member of the executive board of World Federation of Parasitologists. The World Federation of Parasitologists is like an umbrella that actually hosts societies rather than individual members. And one of the activities of the federation is to organize what is called ICOBA. ICOBA is the International Congress of Parasitology. And I like to think about it like someone who was born in 29th of February. So do not celebrate it yearly, just once every 4 years. And it's coming back again this year in summer of 2026. Why it's important to attend meetings? I think it helps to collaborate and to know scientists from different disciplines within even the parasitology field. Like, for example, in this photo, I met the speakers for today's webinar in the International Congress for Tropical Medicine and Malaria, which happened in 2024 in Malaysia. With no further delay, I would leave you now with the first speaker, Dr. Sadura. She is Associate Professor in the Faculty of Veterinary Medicine at Chiang Mai University University in Thailand. The floor is yours and I'll stop sharing.
Okay, so hello everybody. So can you hear me properly? It's okay, right? So it's my great pleasure today to be with you to sharing the Leishmania martiniquens infection. This species of Leishmania may be quite new to many people, also scientists, but in Southeast Asia, especially in Thailand, and in some area in Brazil or in Europe, may be familiar with this one. So today my topic is about the Leishmania multiplicaris infection. I will review in the disease, epidemiology in the humans and reviews in animals and some updates in the diagnostic. So let me review a bit on the leishmaniasis. So leishmaniasis is mainly caused by the parasite we call the Leishmania. That is a genus that is belonging to the Kinetoplastida or family Trypanosoma together with the Trypanosome. Leishmaniasis is quite neglected of the disease. in many areas, especially in our region. So nowadays there are three subgenera of the Leishmania causing the disease in human and also in animals. We may familiar with the Leishmania venia in the New World, and the new one is the Leishmania bunninia, or the former name is the Leishmania ellisii complex. That is just established or found since in 2014 in the Martiniquens Island. The first case of the martiniquensis is in 2014 in the Martiniquens Island. So there are two forms of the parasite. Amasigote, this is the intracellular parasite found in the vertebrate host, and the elongated one or facetated one, that is the Parasitic, that is the insect vector. Leishmania, the Muninia subgenus, we found that it's quite, in fact, from the phylogenetics analysis, we found that it's like the ancient lineage compared to the others, like the Leishmania or Leishmania venia. And some scientists have explored and found that they may have some deep evolution position, or the parasite is a kind of the co-evolution between the host and the insects. But nowadays, the Boninia parasite is emerged, not is then there is a lot of parasites that maybe I will show in the next slide. About the life cycles of the Leishmania parasite, there's two hosts composed in their life cycle, the vertebrate host, that we found the amasigote form, and the vector, especially in the female insects. Up to now, previously we know that the Phlebotomy sandfly is the main vector, but nowadays belonging to the Mundinia parasite or Leishmania, we found that the Culicoides or Ceratopogonids, miting midges, is also can play like the potential vector that already proved in the next slide I will show you later. So in that life cycle, when the infected flies sucks the blood from the host, the Parasitic will go through the blood circulation, and then firstly phagocytized by the macrophage, and then they transform from the Parasitic to Amasigote, and then they multiply inside the cell. This cycle is repeated, and then clinical size depend on the where parasites have proliferate. For example, the cutaneous, we found that the parasite is proliferate in the mucus, in the macrophage, in the cutaneous area. On the other hand, if for the visceral form, the parasite prone to go to the macrophage in the liver or the spleen or the lymph node or bone marrow. That is different kind that it depends on the parasite species and its tropism of the infection, and also we found some geographic distribution that difference, and some explanation is come to like the potential vector that is different from each area. So the clinical form or manifestation of the leishmaniasis, we can find that the cutaneous form, that's this picture, mucocutaneous is in area have the interlink between the skin and the mucosal area. The visceral form or Kala asa, that is called the visceral leishmaniasis, and then the post Kala asa that mainly occur after the visceral leishmaniasis is get better or already ligation, but suddenly or finally they will occur after that. So we call that the post-Kala asa. And the map, upper map is show the visceral leishmaniasis, and the lower one is the cutaneous and the cutaneous. That we found the different species and also the different geographic distribution. How about the burden? The burden is still high compared to the before, like the 10 years before. Nowadays, the new case is less than before. So approximately 1 million new case of the visceral leishmaniasis worldwide, and if talk about the cutaneous leishmaniasis, it's about 1 million case also. So it's still high of the incidence of the disease. And how about the disease in the free zone area? Before, long time ago, Southeast Asia, together with like the Pacific area, we found that we not found leishmaniasis. Leishmaniasis have never been plotted in the Leishmania disease map, but since 2015, the WHO or WOA nowadays is already plotted that the Thailand also is one of the countries which have the endemic of the leishmaniasis, both the visceral form and the cutaneous form. The diagnosis of the leishmaniasis, mainly we have to first can done by the clinical examination. We can see like the cutaneous ulcer, maybe local or localized only one nodule or one ulcer, or the multiple nodules that can be observed. Together, maybe have some fever, some anemia, or liver or spleen enlargement in the case of the visceral form. Another finding that is noisier is like the lymphadenopathy that can be found. The second diagnostic method is like the parasitological way that is more is precise to see the amazigote form. That is we first screening like the leishmaniasis. But nowadays, we also develop the molecular one and then the serological one to diagnose the infection. Molecular is more robust method that can give the precise result of the detection. However, sometimes the molecular or DNA detection also cannot work. So the last method is the serological way to detect the antibody level in the samples, or whether the serum or the plasma. For example, the DAT or the SIP test. However, this serological is the method to diagnose like the indirectly or to review the previous infection. And the another thing that people like to confirm the infection is to do the cultivation or culture. That is more confirmed to the parasite is be already in your specimen. Go to the Leishmania martiniquens infection in human and animal. So I have to say that martiniquens is first found in the Martinique Island, that in patients that have some HIV co-infection. And that guy show like the disseminated cutaneous leishmaniasis form. In that time, the scientists didn't know whether what species is, and finally it's come out to the new species. However, they already classified not belonging to the Subgenera Leishmania or Vienia, but it's belonging to the new one, it's Mundinia. And then in the other area, we also found the autochthonous leishmaniasis cases increasing, especially in Switzerland, in Germany, and also in Southeast Asia, especially in Thailand. And this is the distribution of the martiniquens of the Mundinia parasite worldwide. That this quite new publication, just published in March this year. So they plot the infection. The yellow, the green one is the martiniquens that found almost worldwide, and not only in the animal, or we also found in the human in many areas. So Here, another thing is the vector, not only the sandfly vector, but biting midges like the Qrophides can also serve like the potential vector for the pneumonia. The clinical symptom in a human infected with the Leishmania martiniquens is depend on the immunostatus. So many cases that have the strong immune or we call immunocompetent case that show like the asymptomatic, no clinical signs. However, in some immunocompromised or weak people, some cutaneous form or visceral form, or even in the very weak, for example, who infect with the HIV infection, the patient after infect with the Leishmania martiniquensis prone to present like the disseminated cutaneous leishmaniasis together with the visceral form. So it means such type of this lady also have the multiple or disseminated cutaneous leishmaniasis or ulcer in her bodies and together even in the mucosal area. And also found like the hepatomegaly and splenomegaly too. That is quite interesting for what is the molecule or what is can explain such type of the severe manifestation of the martiniquensis. Distribution of the martiniquensis infection in human in Southeast Asia. Up to now, there's 2 countries that present. So first, Thailand. There's many cases nowadays, and even this year, every month we have the reported case. And just last month we found one case in the northern area, in the Pear area. It's this province. So it means if we observe more, we can find more. And the another country is Myanmar that is found in the patients is they residing in the Yangkung area. So approximately 3 cases already published. Then in the some case is in the healthy patient didn't show any clinical signs like the asymptomatic one, but in 2 cases, that one is the HIV co-infection, the another one is like there some immunosuppression due to the steroid treatment for 2 months. So in that guy, like the picture here, he present like the multiple erythematous nodules, like this figure. Regarding to martiniquensis infection in the animal, we found some cases nowadays. So mainly there's 2 species of the host. So first is the cattle, the another is the horses. So in cattle, the Professor Loftiger is found in Germany, in Switzerland. That the cattle show the ulcer in the different area, especially the area that's in the, like the muscle, in the knee, in the joint area, or in the other. And they found like a martigo. And this case also confirmed that it's a martiniquensis. In non-horses, the case also found more and more. And recently is the case in 2022. So first we found there's a published found in the German and Switzerland. It's a 9 horses got infection and mainly show the clinical signs. There's some strategy to manage with these 9 horses. So half of them got the surgical removal, so surgery, and found that after surgery and they show no recurrence. Some didn't do anything but spontaneous vacation. For example, this horse in Germany or in the northern Switzerland. And however, there are some horses that still get infected and show the recurrence. That is, I highlight in the pink here. But anyway, so this is the evidence that we found in the horses more and more, and in many areas. So how about the leishmaniasis in animal in Thailand? So this finding, we also after we have the case in the human, the outbreak investigation have ever been performed, and then by DNA or by PCR or nested PCR, we found the DNA of the Leishmania martiniquensis in the back lab in two areas, in the Chiang Mai, in the northern here, and in the Songkhla, in the southern. So at least we found that back lab could be served as a potential reservoir. However, in the other uh, animals, for the investigation at the same time, we found only the serological positive, not the DNA positive yet, including the buffalo, the cattle, the dog, and the cat. However, it can be explained that probably these animals have ever been infected. But anyway, we didn't find the clinical signs, symptoms, even in the cattle or in the other species. However, recently, last 2 years, that I found uh, the case in the dog. So this dog is just kept with there's a French bulldog, uh, not old, it's a 5-year-old, no underlying the good health, no disease, confounding with her condition. A little bit fat, but no more other abnormalities. So these animals show only the one nodule in the ear pinna here, ear pinna, the right side here, that's very small. However, the owner worried whether it's going to be like the tumor. So the owner brought him to the hospital. And then we correct the vet, the veterinarian correct the samples. Okay, the veterinarian correct the sample by needle FNA, fine needle aspiration, and we check for the amasticode form and do the cultivation using the gas insect media, enriched with 30% fetal bovine serums, and then after that we also perform the molecular identification with the various techniques. So this is the parasite that we found. So first figure from the FNA, we found the organism like the amasticode form here, and we could success to the cultivate in the gas medium. And after we do the molecular, we found that it's be like the martiniquensis confirmed by the PCR of the mkDNA and the real-time PCR to compare the melting curve analysis. And by phylogenetic confirmation, this case also belonging to the martiniquensis. These dogs have been followed up for like almost 1 year with a different plan to of the approach. So first plan, we didn't give any medicine because we have the previous review that the horses can be self-replication. So we just observe because it's very small. However, it still be there, cannot replication by themselves. So we start to treatment. This is a symbol of the what we done. So we correct the blood samples, periodically, and then we collect the sample of the serum. We also sometimes do the X-ray or ultrasound to check whether the liver or the spleen enlargement occur or not. However, from this correction follow-up, and then we found that no evidence of the visceral leishmaniasis involvement. So it means no anemia, no enlargement of the liver or spleen, or even the blood chemistry showed no remarkable lesion. The treatment that we decide for this dog because it's only the cutaneous and localized area, so we decide the intralesional injection. The drug available in Chiang Mai is like the amphotericin B. So we select this drug. The another reason that we select amphotericin B because it already has studied that martiniquens in Thailand is sensitive to the amphotericin B. And the another application that we have prepared is like prepare the ointment for the topical because after we tried now through injection, the dog was okay. He was pain and rejected for the infection. And this is the treatment outcome. So in the first phase when the case comes like this, and the first phase that I just follow up to see whether it's the spontaneous rejection or not. About 4 months is no spontaneous rejection. So we decided to treatment. So first we do the intralesional injection around this area. So you can see whether the lesion is from the nodule localized change to the multiple nodules. Even it's small, but it's spread out. In this phase 3, we inject for 2 times, and it look good. However, 1 month later, it's still recurrence. So in phase 4, we try the continuously treatment using the ointment daily of application for 2 months.
Dr. Sadura, just the time is almost up. If you
okay.
Thank you.
Okay, so I will give quick. So this is it's almost worked out, but however, 1 month later it's a recurrence. And the last, so we come back to use the intralesional injection and continuously for treatment. And this time it was successfully killed. So, give the quick. So this is some update on the potential vector of the Leishmania martiniquens that found that the kiwicoides could be the potential vector by two evidence. So first one, more important, is the experimental transmission used in the laboratory of the kiwicoides sonolensis. That's it in the lab, and then do the experiment and found that it can be growth of the parasite inside it, and also found the DNA of the martiniquens or other monomia in the biting leaf using the nanopore. There's two evidence that already published. So this is now of the potential vector update for the Leishmania martiniquens and others. So about the S1 that we already there's some developed is first like the quantitative real time. And then the next gen sequencing, especially nanopore technology, has been done. Lastly, the nucleic acid lateral flow or NAFA also developed. However, it's not been in the clinical use yet. And this is the come up to the almost the last slide. That is the mid for diagnosis, serological detection. This is one method to survey the subclinical evidence of the infection. So there's some study mentioned that the recombinant canesin RK39 of the martiniquens can also be produced and have some high sensitivity and specificity. So now we try to expand the epidemiological investigations, not only in the human, also in the animals and potential vectors in our area, and the development of the point care diagnostics also for the animals. And now the team of my and the PhD students, we are developing the test kit, the rapid test for the serological screening of the martiniquens infection. So I think that's all of my talk. Sorry for time because I prepared for 25 minutes. But anyway.
Yes. Thank you very much. Quite interesting, actually. I have a couple of questions in my mind, but I think I would keep them to the end, and I will introduce the second speaker, Dr. Elshafie Hassan. I said I met both speakers in Malaysia, but that's not quite true. Dr. Elshafie since we geographically working in Oman, so quite easy to meet him, and he's always my go-to person when I need any help with blood parasites. I will allow him now. I'll give him the floor to present Trypanosoma evansi. The floor is yours, Dr. Shafir.
Okay, thank you very much, Dr. Hissa and Dr. Surada, for your nice presentation. And good morning, good evening, or good afternoon, wherever you are. And just today, I would like to share with you general an overview about the Trypanosoma evansi infection in camels. So epidemiological background and what is the present situation in Oman. And my name is Dr. Elshafie. I am originally from Sudan and now working in Sultan Qaboos University, College of Agriculture and Marine Science, Department of Animal and Veterinary Science. So I will start give you a brief about trypanosomiasis. We are in the same page with Dr. Surda regarding the protozoa kinetoplastida. So this is a disease caused by a parasite belonging to a genus Trypanosoma. And this parasite could be found in the blood plasma or tissue fluids. And it transmitted by certain vectors. So depending on the vectors, so it could be found the geographical location. So this parasite is transmitted mainly by biting flies. So we could observe that in the Glossina or Sassafly in Africa. So that one, it would be only in Africa due to the presence of the biological vectors. However, we have a certain type of the parasite that it could be transmitted mechanically by Stomoxys tapanos. Whereas in Latin America and South America, there is another type of biological vector that belong to the pox. Another type of the parasite that one in the Trypanosoma could barely mainly on the horses, it could be transmitted by venereal contact or during the mating. So for any biological trypanosome, for any biological vector for the trypanosome, they could be undergoing 1 or 2 cycles of the development in the vector, as we could see for the African trypanosomiasis. Just a brief about the classification for the genus of the Trypanosoma. So they are belonging to the order Kinetoplastida. So they are a flagellate with the kinetoplast. And the genus Trypanosoma, it could be classified into 2 sections. We have the Silverian and we have the Stercorarian. Stercorarian, the infective stage will be in the feces of the vector. And that one, it only be found in Latin America, where we have Trypanosoma cruzi, that it could be a zoonotic for human and also for the dogs. On the other part, we have the Silverian, where the infective stage for the final host, it will develop into the mouth part of the salivary glands. And we have a different subgenus. So we are mainly focusing about the trypanosomes, where our parasite, Trypanosoma evansi, is sharing with other species such as Trypanosoma equipedia that transmitted during coitus. We have Trypanosoma brucei gambiense, Trypanosoma brucei iridescense. Both of them, they could infect the human, only a human. And we have the Trypanosoma brucei that it could infect the livestock animals. So this table just summarizing the different type of the trypanosomes that it could be found in animals. And since in this area, in the GCC country, where we didn't have any sassafly, so we will focus mainly about 2 parasites that could be mechanically transmitted. So we have the Trypanosoma vivax that it could adopt for biological transmission by sassafly or mechanical transmission by Staphanos, Stomoxys, and other biting flies. And we have for sure the Trypanosoma evansi that adopted totally for the mechanical transmission. Therefore, later on, we will see the spreading of the disease is worldwide. So, as we mentioned, we have several species. So for the Trypanosoma brucei, Trypanosoma brucei rhodesiense, and Trypanosoma brucei gambiense, all this group they consider as an African trypanosomes because they are need a biological vector, and in this case it will be the Glossina or the Sisifly. So Trypanosoma brucei, it called the disease, we call it Nagana, that could infect the livestock and the wildlife. And rhodesiense, it will be found in the East Africa, and Gambiense, it will be found in West and Central Africa. And both of them, they call them the sleeping sickness, because at the end, at the terminal stage of the disease, there will be a coma. So if we could observe here, this is a winter pattern sign that it could be observed in the human. Later on, you could find or recover the trypanosome from the lymph node aspiration. Other species that infecting the animals, it will be Trypanosoma congolense. Also, it need a biological vector. We have Trypanosoma vivax, it could be both. Equiberdium during mating, Trypanosoma evansi or Sura. The name Sura is coming because the animal will be very emaciated. It is an old name that found, and it terms from India, telling about that very emaciated animals. So now we will come to the Trypanosoma evansi. So Trypanosoma evansi, the protozoan hemoflagellate and infect a wide range of the animal hosts and giving the reason for the disease, we call it Sura. So the range for the Trypanosoma evansi or the host, it could infect any animals. And as we mentioned previously, it belongs to the subgenus Trypanosoma, which including Trypanosoma brucei, Trypanosoma brucei rhodesiense, Trypanosoma brucei gambiense, and Trypanosoma equivalent. The main issue is that here, the most of them, most of these parasites, they are morphologically same. So sometimes it could be very difficult for us to do the diagnosis by doing the conventional method or to some extent for molecular test. So for the Trypanosoma evansi, they found that horse, dogs, and camels, they are considered as the most susceptible to the T evansi. So the disease manifested by a severe outcome. If the animal is not treated, it could lead to the death. And there is a different strain virulence of this parasite, comparing between the Latin America, between Asia, and North Africa, in the Middle East. So it depends also the host susceptibility, because this disease, also if there is any stress or the immune system is compromised, that one it will lead to the exaggeration of the infection. So this parasite, it will cause fever, anemia, emaciation, edema, and anorexia. And at the terminal stage, it could develop neurological symptoms that also abortion, it could be observed during the late stage. So here we could see that how this horse is emaciated. This is a photo for a horse that previously we injected with the Trypanosoma evansi to study a certain variant service like a protein. And corneal opacity also, it could be observed in the dogs. So the economic losses, it could be related to the mortality, chemotherapy cost, reduction in the milk or meat production, infertility, and also abortion. So the Trypanosoma evansi adopted mechanical transmission by tetanus, hematopoietic chrysops, and estomoxis. So it is just like a syringe passage. So you get the infection from the infected animals, and then it is transmitted to the healthy animals. And this one, it will take only a few seconds, where the parasite, it will develop by the binary vision. So the morphology for the Trypanosoma evansi, which is very important for us for the diagnosis, is considered as a monomorphic. So it has a cylinder form that with the length between 25 to 35 micron, free flagellum, undulating membrane, and most of the time, the nucleus, it will be toward the interior parts. So this is a direction toward the free flagellum. So the free flagellum, it will pull the body of the trypanosomes. Because later on, we need to know This is the same morphology that could be observed by the Trypanosoma brucei, and that it had another form of intermediate cylinder form or stumby form, where it is uncommon in the Trypanosoma evansi. Another route of transmission could be found for vampire bat in South America. And the most interesting part about the Trypanosoma evansi is that in 2005, there is a case in India regarding the Trypanosoma evansi, a patient who's an indolating fever developed, and then they did the test and they found that this patient or this guy was infected with the Trypanosoma evansi. And when they did the investigation, they found that there is a certain type of the lipoprotein, they call it APOL1. There is a mutation there, and that protein, it will be found in the plasma. So if a Trypanosoma evansi enter a human body, it will lead to the lysis of the trypanosome. And due to the mutation of that lipoprotein in that patient, that one, it leads to the development and the propagation of the trypanosomes in the blood of that patient. However, in 2015 or 14, there is a cases from another areas in India, from Vietnam. They found that there is a patient there. Also, it was infected with the Trypanosoma evansi, and the APOL1 lipoprotein, there is no mutation. So the only history that the patient delivered before 2 to 3 months. So that one, it opened the door to either Trypanosoma evansi, if there any evolution in that parasite to become a zoonotic. So we didn't know so far because of the lack generally of the funding for studying the Trypanosoma evansi, because it is thought that it is only could infect the animals. And here you could see the tapanos, as we mentioned in the mainly, and istoboxes, they are the mainly biting flies that they help for the dissemination of the parasite. However, we have a carnivorous. They found that if a host, like a fox eating a meat of a rodent that is infected by Trypanosoma evansi, there is a chance also for the transmission. So the distribution of the Trypanosoma evansi, it is, as you could see, it is more focusing on the Latin America, North Africa, in Africa, Sub-Saharan Africa, and also in the Asia, Russia, and also there is a sporadic occurrence of the Trypanosoma in France and Spain from camels imported from Morocco through the Canary Islands. So the main idea is that Trypanosoma evansi originated from Trypanosoma brucei, and the Trypanosoma evansi lacks the maxicircle kinetoblast DNA, and that is a marker it used later on to diagnose the Trypanosoma brucei. So by lacking that maxicircle kinetoblast, it allowed the Trypanosoma evansi for mechanical transmission. Therefore, the Trypanosoma evansi expanded beyond the Sessi belt, where it is only in Africa. The most interesting part regarding the Trypanosoma evansi and Trypanosoma brucei, that they have what we call it the variant service glycoprotein. And this is considered as the physical barrier of monolayer glycoprotein that covering the whole of the trypanosomes, and they provoke the host immune system. If you could look to this graph, so the breast image will get hot. That one, it will provoke the immune system. Immune system, it will produce antibody to fight that peak of parastemia. Then the number of the trypanosome decline, and once it is declined, then it will change the outer coat. And that is the main idea of the variant service glycoprotein or intergenic variation adopted by the Trypanosoma evansi to escape or to evade the host immune system. And there is an order of the expression of this variant service glycoprotein, where there is a certain protein will be expressed early during the acute infection. And the region of this protein It is about 400 to 500 amino acids, and it has a variable or the N-terminal domain. Most of the time, it will be the highly variable region because recognized by the antibodies, whereas the C-terminal, it will be conserved. And that is why we could not find a vaccine for the Trypanosoma evansi because of this variant service glycoprotein, because they found that more than 1,000 genes of the Trypanosoma evansi and Prusii, they could coding for that variant service glycoprotein. However, they found that there is a certain type of the Trypanosoma evansi that lack this ROTAT 1.2 service glycoprotein. So ROTAT 1.2 service glycoprotein, it expressed by the Trypanosoma evansi only. So it is not expressed by the Trypanosoma prusii. However, there is certain type of the strain of the Trypanosoma evansi, they lack that ROTAT 1.2. And later on, we will see, because it will be used for the diagnosis, specifically for the serological test. And that isolates that lacking the ROTAT 1.2, it found in Kenya, Ethiopia, Chad, and Sudan. So in this graph, it was published in 2018, and during that time, we did an experimental infection in local ponies, in certain ponies in Malaysia. And we found that the indolating prostemia that is going. So the infection was almost for 1 month. So we recover about 4 peaks of a totally changed variable surface glycoprotein. And most of the time, the temperature, it will follow the prostemia in the blood. For that reason, it is very important to collect the blood during the high fever in order to recover the parasite from the blood, because it will be abundant in the blood. It could reach up to 10 to the power 6 per 1 milliliter of the blood. So for the diagnosis, for the T. evansi, it is not straightforward. So there is a different level of the test that we could implement it. So based on the sensitivity and the specificity of the different type of the test. And so the routine one, we call it the traditional parasitological examination, where we could see the parasite. And that one, it could be done by thin or thick blood film. It could be enhanced by making the hematocrit centrifugation technique, HCT technique. It is a more sensitive compared to the first one. And finally, we have the mice inoculation. So if we collect the material from a suspected animal, inject it into the mice, then there will be a propagation of the Trypanosoma evansi. The good thing about the HCT is that it will help us to identify the PCV, because Trypanosoma evansi, it will lead to the hemolytic anemia. So anemia is a common finding, specifically during the chronic infection. We have serological techniques. So there is a many antibody detection tests, such as ELISA IFA. The most recommended one by the OIE was the CAR agglutination tests for T. evansi, developed by the Institute of in the Antwerp in Belgium. And that is a very rapid test that it could be used for, also it could be used in the field. So it was developed by the Institute of Tropical Animal Health in the Antwerp. And also there is an antigen detection. However, that one lacking the sensitivity to some of the antigen detection. And the problem with the antibody detection that they could not differentiate between the post and the current infection. For that reason, we could not make the judge for the treatment based on the serological test by antibody detection. For the molecular techniques, we have different techniques. There is conventional PCR. We have a different primer, such as ITS, ROTAT 1.2. Actually, ROTAT 1.2 is specific for the Trypanosoma evansi because it was not expressed by the Trypanosoma prusii. We have another TPR, and we have a non-ROTAT 1.2, and that one, it was developed actually for detecting the non-ROTAT Trypanosoma evansi. We have the lamp nowadays, it is developed, and also we have RPA, but there is still advantage and disadvantage, and also the lab equipment. It could affect the use of the different molecular techniques. So for the direct or the conventional, there is a sensitivity issue that it is recovered by different application, that there is a different type of the primers it could be used. However, the sensitivity is questionable, where some of the authors recommend to use a TPR as a more sensitive compared to the others. However, for example, ITS1, it could help to identify the species by doing the sequence and retrieve the sequence from the gene bank to identify the isolates. So control and the treatment, there is a many trypanocidal drugs that it could be found. We have the suramine drug, and this is the oldest drug, and Trypanosoma evansi develop resistance. We have the isometamedium chloride, and that is a very effective against Trypanosoma vivax. However, some also argue about the effectiveness against Trypanosoma evansi. We have the quanapyramine sulfate, which is good to be used for the treatment of the Trypanosoma evansi, but they observe in the hordes that they would be a relapse after a few months. We have a diamazine acetate. It is used also in the ruminants. It is sensitive for the to be used for the ruminants, and it is massively used in Asian, Southeast Asian country. So there is also a report of developing a resistance to it. And we have the similarisan. This is the most recent safe potent drug that could treat the Trypanosoma evansi infection in buffaloes, camels, and cattle. However, there is still a questioning about the resistance. If the Trypanosoma evansi develop a resistance to it or not, because generally there is a massive use of the trypanocidal drug by the animal owner, specifically for those who find in the remote area. So they could do also for the control quarantine measurement, and there is a nice successful story in the Canary Islands about quarantine measurement extended almost 2 years in order to control for the camels infected by the Trypanosoma evansi. Or we could do the vectors control, and this one it could be handled by the trapping or doing the sound management. However, in the most of the case for the vectors, to control them, I think it is not an easy task. So now we will dive to the what is happening in Oman. So we will study, or we will cover some research that conducted in Trypanosoma evansi in Oman. So Sultanate of Oman is characterized by arid climate and sometimes semi-arid in southern part of the country or the mountainous region, and it has a very long, about 3,165 kilometer coastline along the Arabian Sea and the Gulf of the Oman. And the total population of the camels, it is roughly around, it is 273,000, and that one it was in 2019. So Oman is bordering, or Sultanate of Oman is bordering Saudi Arabia, Yemen, and the United Arab Emirates, and it's composed of about 11 governorates that they are composing of different wilayats. So here the surah, they call it Zabur, and it was first detected in 1982 in a camel that's suffering from anemia, lacrimation, pyrexia, in emaciation. In the photo here, I have, this is my student, my friend, Mazen Kharasi, conducted his PhD, who conducted his master degree under my supervision in the Trypanosoma evansi in the camels. So the disease is reported in camel in the government and the private clinic, so there is no scientific publication. And it was reported that TB bats began to spread to Asia because it is imported livestock from Africa. So And in Oman, so far, there is no Trypanosoma vivax. However, there is a report in the neighboring country, specifically in Saudi Arabia and in Iran, that they detect the Trypanosoma vivax in the camel.
Excuse me, Dr. Elshafie.
Yeah, I'm about.
About.
Okay, no problem, Dr. Hessa. Yeah, now it is almost 23 minutes, so I hope it's wrapping up.
Yeah.
Okay, so in 2000, this publication, this is the first publication that found after the detection of the trypanosome in the camel, and that publication was in 2020, and it was conducted by the Central Laboratory of the Animal Health Ministry of the Agriculture and Fisheries. They received a blood sample from different governorates, so they collect about 500 blood sample during 2017, 18, and 19, and they did a microscopic examination. They found that the prevalence by doing the traditional parasitological technique in 2017 was 0.54% and increasing to become 7.25% in 2019. And the total length, they did the more phometric measurement, and they found that the total length, including the free flagellum of the Trypanosoma evansi, is 24.9. After that, be with my PhD student, we try to develop a sort of a retrospective study. So, because here they have in the central veterinary lab, they have a sample bank for the serum. So we found that there is a 4,364 serum sample between 2014 and 2015. So we applied the carb agglutination test for the T evansi, as we mentioned. The rapid tests are relying in the ROTAD 1.2. So the overall seroprevalence was 38%. So it was a retrospective study. After that, we conducted a cross-sectional study in the 5 regions in the northern Oman that including Ibrahim, Al-Batna, Al-Dakhiliya, and Sharqiya. And we collect 388 camel sample, and we run the thin blood film where the prevalence was 0.8%, by carb agglutination test was 24%, and also we applied the TPR for the conventional PCR, which was 77%, and ITS 31.6%, and ROTAD 1.2 was 8.4%. Then my student Mazin, he did his MSc, and we collected between 2020 and 2021 425 blood samples from Al-Batna. We focus on Al-Batna governorate, composing of Al-Batna South and Al-Batna North, and it was bordering with the UAE, where there is a high density of the camel in that area, and also it is near to the Masbah, to our lab, where we could do the hematocrit centrifugation technique, because the parasite, it will die within few hours. So the life is dependent on the glucose in the blood. We found that the overall seroprevalence was 19.5 in that area. And the risk factor, we found that tick-free camels, racing camel, and camel less than 5 years old show a high Trypanosoma evansi seroprevalence than other camels. So for the location or the wilayat within that area, we found that in Al-Musana'a, we get a high prevalence of it was a highly seropositive, and that one, during the questionnaire, we found most of the people in Al-Musana'a, they keep the camels with other animals, other ruminant, that they could act as a reservoir. Whereas in Al-Khabura, we found the prevalence, it was 10.5, which is less compared to Al-Musana'a, where the people in Al-Khabura, most of them, they keep the camels separate from the others. So this gives a suggestion that it could be mixing the camels. It could lead to the spread of the surah in that area. Then after that, for the 425 plus sample, we did the blood film, we did the HCT, and after that we do the conventional method. We use a very sensitive primer, TPR 1 and 2, and the positive result from that primer, we apply the ITS 1 to characterize, isolate, and we applied the Rotat 1.2, and we applied the Rotat 1.2, and also we check for a certain specific primer for the Trypanosoma vivax. So here you could see the variation on the prevalence by using the blood film. This is a direct smear thin blood film, and this is a hematocrit where we could see the movement of the Trypanosoma under the microscope, and the TPR where the prevalence was 13.9. Between the location, both north and south, there is no significant. However, there is a high prevalence generally in the Alpatna South, where it could be linked to the mixing of the camels with the other ruminant in that area. So the TPR, we recovered 59 samples. We did the ITS, we found 15 positive. We sent it for sequence. And this 95, we run the Rotat 1.2. So we get 55 positive sample, whereas 4 samples were negative. So we saw that it could be non-Rotat. So we ordered the primer and we apply on that 4 sample, and surprisingly was positive. For the Trypanosoma vivax, we didn't recover any positive case. So just here, it is indication for the risk factors that it could be observed. And most interesting here regarding the herd structure, despite the fact that it was not significant, but still, once the camel is mixed with the other ruminant or other animal, there will be a high prevalence. Here we have the BCV and the total protein. We could see that it was high in the positive case, so they could be used as a marker later on to be confirmed for the infection with the Trypanosoma evansi. For the ITS, we found that the Trypanosoma evansi sequence, it is a close related, which is a 97% with the water buffalo, the domestic water buffalo in Thailand. And all of our isolates, they have a close similarity of 96.3 to 97.8. And then for the Rotat, we also do the sequence, and we found that it is highly similar, which is a 99.2, with the isolates from camels in Kenya. For the non-Rotat, unfortunately, we didn't find an isolates or sequence in the Qingpang, so we didn't have phylogenetic for the non-Rotat 1.2. So conclusion, based on our research, the Trypanosoma evansi, the only Trypanosoma species present, no Trypanosoma vivax. So camel deviated of tick infestation, because tick infestation it is related to the pyroplasm, young animals, and leisure riding camels. Under 5 years, they are at high risk for contracting the Sura. And the PCR, by using the TPR primer, is more sensitive for detecting the Trypanosoma evansi in the region compared to the microscopic examination, and also the cardiac agglutination test for serology. So reduction in PCV, elevated protein could be an indicator for Sura. And the most important part is that this is the first detection of non-Rotat 1.2 in Oman. And that one, it will open the door that for the usage of the cardiac agglutination test for Trypanosoma evansi, because it will show a false negative when in reality it is a positive, but the isolate is lacking that gene. So this is an acknowledgement. We thank Sultan Qaboos University for the grant, staff of our department, Animal and Veterinary Science, and also the staff from the Animal Health Research Center, the Minister of the Agriculture, and Al-Falah Private Clinic for helping for the sampling. And thank you.
Thank you, Dr. Elshafie. Thank you very much. I'll go back to closing remarks. Then I will stop the recording and we can have a discussion and question. I hope Dr. Saroda is still with us. Okay. So this is the upcoming ICOBA, which is in August, is the 16th to 21st of August, and it will be in Montreal. And the leading person is the first Vice President, Muammar Nadaw. And the registration is still open if you wish to join. And here is the more detail about the event. We started the series for the road to ECOBA, and the third one would be in a month from now, and it will be by Professor Linda Eva Amoha from Ghana, and it will also coincides with the Malaria Day on 25th of April. More announcement about that would come later on in the website of World Federation of Parasitology. I'll end now the recording. And before that, I want to thank both the speakers and the participants also who attended here. Thank you very much.